Welcome everybody to this week's edition of the Eevee Journal Club. Thank you for joining, and we have a very special presentation today by somebody who's been working with schistosoma EVs for quite some time, and is probably one of the world's leading experts on this organism and the vesicles that it releases. So Marije Kuipers is coming to us today from Leiden University in the Netherlands. She also is co-appointed at Utrecht University. So she, when she can, she goes back and forth and does different experiments in different places. So she's kind of an itinerant scientist, and she's now in the final year of her PhD. So we will have the chat box open during the whole presentation. Please make your comments and ask questions there, and then at the end of the presentation, we will open the mics so that you can ask your questions, and we look forward to a good discussion. So Marije, thank you so much for speaking with us today, and I'll hand it over to you.
Thank you very much for the introduction, Ken. I will start sharing my screen. So yeah, as Ken said, I've been working with Schisto and EVs for quite a while. So I was actually really happy that I finally got to share it with the world this year with publication in the Journal of Extracellular Vesicles. And I will start by telling you something about the Schistosoma mansoni, the parasite itself, and the complex life cycle. So you might have heard of schistosomiasis or bilharzia. It's actually endemic in the tropical areas of the world, and over 200 million people are infected or at risk. And so it is endemic in the tropical areas because of the intermediate host, which is a snail, which lives in the fresh water there. And from the infected snail, cercaria are released, which then can penetrate the skin. And upon penetration, it loses its tail and transforms into a schistosomula. And these schistosomula then move through the body in the blood vessels, via the lungs towards the portal vein near the liver, and they then are mature worms, the adult worms, and the males and females will form pairs, which would then start laying eggs in the vasculature around the gut. And the eggs can then be expelled by the feces, and upon reaching the water again, they hatch, release a miracidium, which can then reinfect the snail host. And the interesting thing about this parasite, so our department studies the parasite not only eventually to develop vaccines, for example, but also because this parasite can live in its host for over a decade. And it can do that because it has established really advanced immunomodulatory properties. And that's also what we try to understand, because if we can understand how this parasite can modulate our immune system, we can actually use the molecules they secrete or the same pathways to also modulate the immune system during allergies or autoimmune diseases, so where during inflammatory disorders. And the immune modulation that is mediated by these parasite life stages is that during the initial phase of infection, so with the schistosomula, there's a mild Th1 response observed, and the eggs actually have been able to induce a really strong Th2 response, and the adult worms have been associated to the upregulation of the regulatory arm of the immune system, also mainly during a chronic infection. And part of these effects have been attributed by the excretory secretory products these different life stages release. And for the remainder of this talk, I will mainly focus on the schistosomal life stage, and this is how they look under the microscope. And personally, I think they're really cute to see them wiggling, and I always love to watch them under the microscope, how they move, and you can really nicely see how they are multicellular organisms. So more about the initial phase and the immunology during this infection of this life stage, which happens in the skin. there's actually a delicate balance between the pro-inflammatory responses by the host immune system of the parasite, but also the already deduction of some regulatory factors such as by immune cells. And the idea behind it is that because of the regulatory immune system, that is the regulatory part of the immune system that's also activated, of the adaptive immune system. And this allows not only the parasite to fully develop into adult worms and start laying eggs, but it also allows reinfection because there's no full immunity generated. And some colleagues of mine have actually published some really nice data on how this skin penetration happens. So they fluorescently labeled in Cercaria and then added them to a skin biopsy. And here you can nicely see how these parasites enter the skin of its host, finding their way to blood vessels. And in that same study, my colleagues also added Cercaria together with monocyte-derived dendritic cells in a well and then looked at immune responses. And they also did that in a trench well system where there was no direct contact between the Cercaria and the cells. And then they also saw that there was an increase in programmed death ligand 1. So that's also a regulatory marker, and also IL-10, again, suggesting that the excretory secretory products are involved, so what the Cercaria release. And another study has been showing that if you take these ES products from transformed Cercaria that were cultured for 3 hours after their transformation, and you add this to whole blood of infected individuals, there's also an increase observed of pro and anti-inflammatory cytokines such as IL-8, TNF-alpha, and IL-10. But if you pre-treat ES with periodate, so that kind of crudely destroys the glycans in this mixture, there's actually a reduction in these responses, suggesting that part of the ES immunomodulatory responses is due to glycoconjugates in this mixture. So what are glycoconjugates? So glycoconjugates are glycans. They are sugars attached to each other in certain linkages, forming certain sugar motifs. And this is an example of the mammalian glycans that can be present. For example, there are glycoconjugates, so glycans attached to a protein, either via an asparagine called N-glycans or via serine or threonine via called O-glycans. But you can also have glycans attached to lipids, called lipid glycans, and for example, also proteoglycans with even longer structures, which you can also think about in terms of the glycocalyx. And these glycans are highly diverse. They differ, the profile a cell can have in terms of glycans, they differ between the status of the cells, the cell types, but also organisms. And the biggest difference that you should take into account when comparing mammalian glycans to the glycans of Schistosoma mansoni is that the worms, helminths themselves, don't have this diamond shape, so the sialic acid glycans in their repertoire. So this is actually what we know from, we have really extensive studies have been performed on the glycans of these parasite life stages, actually done by my supervisor, Cornelis Hokke, and his co-workers. And what you can already see nicely is that there's actually also a difference in glycosylation motifs between the different life stages. So between the Cercaria and the worms. And this is also what they've nicely have shown using antibodies generated in-house targeting these different glycan motifs. So the Cercaria express different glycans than the adult worms and the eggs. And interestingly, these glycans can also be recognized by the host. For example, they have found antibody responses against some of these glycans in infected individuals. But of course, more importantly, and also for this talk, these glycans can be recognized by C-type lectin receptors on antigen-presenting cells, for example, via the mannose receptor, by DC sign, or the macrophage galactosyte lectin. So back to the more like the beginning slide. So we have the ES products of the Schistosoma. We have glycoconjugates in there. There's something with the immune system. But of course, recently, we've also found that, or at least our collaborators in Eevee also published something in JEF, that these parasites release extracellular vesicles. So our question was, do these EVs then also contain glycoconjugates? And do these glycoconjugates interact with the immune system? And do they also have an influence on the immune modulation? So to obtain the EVs, we get the Cercaria. So we have our own infected snails in-house. And I get the Cercaria, transform them to Schistosoma, and then culture them for 3 days. The good thing is I don't have to add FCS to my culture, so I can just grow them, or at least culture them in plain medium with only antibiotics, which saves a lot of hassle along the way. And after 3 days, I collect the ES, do multiple centrifugation steps to end up with an MB-enriched pellet. And as a control, I always take along plain, just medium, also in a flask, also culture it for 3 days, but without the parasites, and then process it or stain it exactly the same way. I actually also uploaded it on the Eevee track if you want more information. First of all, what we did is, okay, do we actually have EVs? So our collaborators in Eevee, they nicely did some negative stain transmission electron microscopy on these EVs, and indeed we did nicely see an EV population between 35 and 190 nanometers. Indicated with the white arrows. The black arrows, they are artifacts that we also observed in PBS only. We also did NTA, and if you have a sharp eye, or what was really weird actually was that with the NTA, we did observe an entire population of EVs between the 200 and 700 nanometers that we did not detect with the EM. So we were like, okay, what happened there? You know, why do we miss something here? You know, did something wrong? Did we lose a population? So the next thing we thought was, okay, let's do cryo-EM, because that gives a better idea of the near native state of these EVs. And I can remember that I gave a sample to my colleague at the cryo-EM facility. And the moment he was measuring, he actually sent me an email like, Marije, you should really come because they look so awesome. It's like they have hairs. So I directly went there, and I just joined him while he was measuring. And what we did see was like almost half of these EVs had like really thin filament-like structures, like hairs on their surface. And which actually hasn't been seen before for eukaryotic EVs, only for OMVs from bacteria that might also sometimes have hairs when they're really highly have LPS or something. It really looks like a glycocalyx. So we took more pictures, and I measured actually the diameter of the EVs, or the size of the EVs, including these thin filaments. And if you compare that, you can actually see that indeed, if you only measure the diameter, you get only sizes up to maybe 200 nanometers. But if you include these filaments, you can actually see that you get kind of the same pattern that we measured with the NTA. And I think this really substantiates that the cryo-EM is really a nice additional value of looking at the true, really the near native state of the EVs that you use. Further on in your experiments or whatever. And so yeah, so that was okay. We have, so for now I cannot tell you actually more about the exact structures that these filaments are. We're still looking into that, but we of course have a hunch that it might be glycans. So the next thing was, okay, now we have nicely isolated EVs. What do actually do they do? Do they interact with immune cells? So we have a protocol that we use a lot in the department using monocyte-derived dendritic cells. So what we then do is we isolate monocytes from blood of healthy volunteers and then differentiate them to immature dendritic cells. And then you can stimulate them. So what we did is took the immature dendritic cells and then incubated them for 2 hours with pKh-stained vesicles, and then measured the cells with flow cytometry for fluorescent. So if the cells get more fluorescent, they interacted or have taken up the EVs. At first we did a dose response measurement. So on the y-axis, there's the mean fluorescent intensity of the cells. And on the x-axis are different concentration of EVs. And of course, we included a diet control to know that we're not measuring pKh aggregates. And we nicely saw there was a dose response in binding or uptake of the EVs by these cells. We also did this experiment on 4 degrees, and interestingly, we did not see anything, any positive cells, almost not positive in this condition, in suggesting that the cells were actively either binding or taking up, probably internalizing the EVs. So we also did confocal microscopy, and that indeed confirmed that these EVs were internalized by the cells. So to investigate whether this internalization or this interaction involves maybe C-type lectin receptors, the easiest and quickest thing you can do is take away calcium from the medium that the C-type lectin receptors need to bind to the ligand. And one of the easiest or most used calcium chelators is EGTA. So we pre-incubated the cells with EGTA and then added the EVs. And indeed, what we saw was if you pre-incubate the cells with EGTA, there's a significant reduction in EV uptake by the cells. And to really know for sure there's uptake and not binding, again, we also did a post-treatment with EGTA, kind of that, and if you do a post-incubation, you will remove all the bound EVs still. So this difference between no treatment and the post-EGTA, that's what the EVs that were still bound and not taken up. And again, we also confirmed with confocal microscopy that indeed there were no EVs taken up by the cells anymore when you pre-treat with EGTA. So we thought, okay, this really looks like the EVs have glycans that might be able to interact with C-type lectin receptors. So let's measure the glycans. So what we did is just started with the N-glycans. Those are attached to the proteins. And you can cleave them using an enzyme called PNGase F. And so we destroyed the entire EV and then treated that with the PNGase F and then just looked at the total glycans that are either on the outer or inside of these EVs. And what we did see, and I know it's quite a complex picture if you're not used to looking at mass spec pictures, but I think the important thing to know is that these are not, so there's no y-axis because these are not quantitative measurements, but it's a relative abundance. So the higher the peak, the more it is present compared to the other motifs in this measurement. And the awesome thing was that this was a similar profile that what we have observed from the extracts of whole Schistosoma. And of course, if you compare it to the ligands that can bind to the C-type lectin receptors, indeed you can see the same motifs in the spectrum as the ones that can be recognized by the mannose receptor and by DC sign. So the next question would be, which of these motifs are actually expressed on the surface of the EVs? So for that, we treated intact EVs directly with the PNGase F, cleaving off the surface accessible N-glycans, and then repelleted with ultracentrifugation, taking the supernatant as for a measurement of the surface N-glycans, and then taking the pellet and then destroyed the pellet, treated it again, and measure that as the shaved, so that the glycans that are most likely on the inside of the EVs. And what we did see that there was actually a really difference between the glycans on the surface and on the inside. So on the surface, we mainly found these complex glycans containing what we call a Lewis X motif. And Lewis X was already known from being present on material from the eggs. It's present in egg material, but also in schistosomal material. And it is known to be able to bind and be recognized by DC sign and the mannose receptor. And on the inside, we mainly actually find these oligomannose structures. So this really seems to be a different glycan profile on whether it's on the surface or on the inside of the EVs. So the next thing, of course, we say, okay, both these structures can actually bind, so the Lewis X can bind to the mannose receptor and DC sign. So can we really find out which of these receptors are involved in interaction with the EVs? So we did a pre-incubation of the cells with antibodies inhibiting these specific receptors on the monocyte-derived dendritic cells. Again, for 2 hours incubation with the EVs, and then measure them with the flow cytometer. And here I have some histograms of looking at the gray plot is actually the cells without EVs, and then the red is when you add the EVs. And then on the we have the DC sign added or block the mannose receptor. We use an isotype control, and of course again the dye control. We also took along DCER, which is actually the dendritic cell immunoreceptor, which does not bind any of these ligands. It actually can recognize Lewis B and sulfa Lewis A, but it has been shown that this receptor is involved and able to bind some of the secarial antigens, or like in the ES. So that's why we took it along. But if you look closely, if you look well, you can actually see that there's no influence on blocking the mannose receptor or DCER on the uptake of EVs. But however, if you block DC sign, there's almost no uptake of EVs anymore. So this is also what you see if you just plot this nicely, because this is only for one donor, but this is of course for more donors. Was really robust finding, and we also did confocal again, also observing that if you block DC sign, there's no uptake of the EVs anymore by the cells. So the next question would of course be, if you treat the EVs with the PNGSF, so if you cleave away all these N glycans that possibly have this ligand to interact with DC sign, do you then also block the uptake of the EVs by these cells? So we shaved off the N glycans and then add these shaved EVs to the monocyte-derived dendritic cells again and measure them by flow cytometry. And interestingly though, so on the left side we have the control EVs that are not shaved, and on the right side we have the data from the shaved EVs. And as you can see, there's not a significant, it's only a really mild, but it's definitely not significant reduction in EV uptake when you cleave away these N glycans on the surface. And interestingly, these EVs were also still blocked in uptake when you inhibit DC sign. So then we look back at, okay, but what are the other glycans these life stages express or can generate? And here are the glycans that can actually attach to lipids. So these are the lipid glycans, and they also have these fucoses that can be recognized by DC sign. So we thought, okay, are these EVs, do they also then contain lipid glycans? So therefore we did a lipid glycan analysis, which you actually have to destroy everything again, but then in chloroform and methanol. So, and then you can, if you have get all the lipids out, you can then also cleave off the glycans using endoglycomer ceramidase. And I think it's important to know, because with this protocol, you can never ever cleave off any, you can never know what is the surface or what is on the inside, because you have to destroy everything to obtain these glycans. However, we did really find a lot of lipid glycans in these EVs, also containing similar glycans actually as we have observed for the Cercaria extract, but in so there are much more higher glycosylated glycans actually. And we haven't tested or investigated how the terminal motives actually look like, but if you look at all the possible motives that could be there, it's probably a mixture. It includes, again, the ligands that can be recognized by DC sign, suggesting that it's probably both the N-glycans and the lipid glycans that contribute to this interaction. So next, of course, we would like to know, what are the immunological consequences of this interaction, and what happens if you add the EVs to these cells. So for that, we stimulated the immature dendritic cells with the EVs for 24 hours in the presence of IL-1 beta and TNF-alpha. If we didn't add this, we couldn't almost detect any cytokines, and we chose these cytokines because they are also present during schistosome infection. And then we collected supernatant for ELISA. And we did see that in the presence of IL-1 beta and TNF-alpha, the EVs were able to augment or form some synergy with this response and increased IL-6, IL-10, and IL-12 production by these cells. And we did also measure the cells with flow cytometry after 48 hours. And then we saw an upregulation of co-stimulatory markers CD80 and CD86. And we also looked at the regulatory markers that I've discussed in the beginning. And there we also did see an upregulation of regulatory marker PD-L1. So then we thought, okay, what happens to these responses if we, again, block DC sign? So we pre-incubated the cells with the anti-DC sign antibody and, again, incubated the EVs. But however, these responses were highly various between the donors and definitely not significant in altering the response. And also for the markers, the cell markers, we didn't get any significant changes when we block DC sign. So we were thinking like, okay, what's happening there? Are the EVs maybe taken up, or is the blockage not successful? So we also looked at the staining of, so the internalization of the EVs after 48 hours. And we did still see that there was a significant, strong reduction in uptake of the EVs when we blocked DC sign. However, if you look at the histograms, you can see that still almost all the cells are positive for the EVs, but just less fluorescent. So this could suggest that after a prolonged time of culture, that the EVs still enter the cells, but maybe via a different receptor or a different route than DC sign. So therefore, we did a shorter incubation. So we only incubated the cells for 6 hours with the EVs, and then isolated RNA for doing a qPCR. And there we did see that there was a significant, at least a significant reduction in TNF-alpha, and also a trend for IL-10 when you block DC sign. So this, especially in the initial stage of this stimulus, it seems that the interaction of the EVs via the internalization of the EVs via DC sign have an effect on the immune response. So to summarize, we isolated the EVs of Schistosoma mansoni, and we show that these EVs contain glycoconjugates, both on the proteins and the lipids that contain ligands for DC sign, and that these EVs and these ligands actually really interact with DC sign on the host monocyte-derived dendritic cells, and that this is partly influencing the immune response by these cells. And I think most importantly to know is that the EVs don't look like how I pictured them here, but they actually look like this. And with that, I would like to thank my supervisors, Hermelijn Smits, Ron Hokke, and Esther Nolted-Hoen, and all the other people who helped me with these experiments. And I also, if I'm kind of made you partly enthusiastic about this, I also have a poster where we looked at the glycosylation of the worm EVs, and I will show that on the ISEF in July, digitally. So yeah, I'm happy to answer questions and discuss.
Excellent. Thank you so much, Marije. That's great. So I've really enjoyed your presentation, and we've had a few questions that have come in during it. I wanted to start out though by just asking you about those large EVs that you were detecting by NTA. Do you think those were the upper limit that I guess you said up to 700 nanometers, or are there some do you think that are even larger than that?
You mean the size of the EV filaments? Ooh. Well, if I'm not sure about the NTA because I did measure over 1,000 EVs with the cryo, and then it would be so large that we also measured them with cryo. So I think then maybe it could be due to the isolation protocol that you might miss even bigger particles that might look like this. But with these isolation, I don't think they are bigger than 800 nanometers, and you do pick them up with NTA. But I must say that what I do with the NTA is that I actually measure on 3 different camera levels. So also the lower camera levels, so you don't measure the small particles, but more the bigger particles in that sense. And then I combine these results and then generalize it from there. So if you only measure at 1 camera level at the NTA, you're going to miss populations of EVs.
Right. Good. Okay. So we have a question from Phil of Askenase. Phil, I'm unmuting you right now, or you can unmute yourself if you want. And you had a question about some of the endosomal escape.
Yeah, this is off of the spectacular demonstration of these huge hairy EVs. But a recurrent finding is that we easily find the EVs in the cells, presumably in phagolysosomes, and they probably resist the acid enzymes there. So I think we have to think about new ways they could be releasing their contents. Now, of course, these could be ones that don't release and so on. But it seems possible that there's a whole new world of releasing from these tough little exosomes that are resisting digestion in these sites, demonstrated by these beautiful fluorescence microscopy pictures. So it's off of the title here, but a general question.
Yeah, I think that's a really nice thought, and I do share the idea that these structures provide some protection for these EVs, because what I also have the idea that because I also freeze thaw them, and it seems like they are also less, I mean, they're still affected by the thawing, but they're less affected by other EVs. It seems like they're protected somehow. And yes, it could have massive implications on what happens intracellularly.
Do they have all the hairs when they're inside of the cell?
I haven't checked it. That would be amazing to do, but then you probably need to either find a way to stain the hairs, if we know what they are, but other thing what might be interesting to do is to do cryo-EM on a cell that just have taken up the EVs.
Yes. Lots to do.
Yes. I need another 4 or 5 years.
Are you going to do another PhD? No.
Yeah.
No, just joking. But so do you? So
Let me stop sharing, by the way.
oh, all right, all right. So the is it necessary to have cytoplasmic uptake though? I mean, do you think there's any role for delivery into the cytoplasm, or are you really seeing this as a signaling event?
And for these EVs, I actually don't know, but I do know that they have tested it for EVs of I think it was Schistosoma japonicum, and they looked at RNA transfer, and they did find some RNAs in the cells, but still I don't know whether how that is delivered. I have honestly don't know whether there's also maybe some fusion or where they end up, if they end up in lysosomes or not.
Mm-hmm.
We haven't looked at that yet.
Great. Okay. So I'm going to ask a question here for Richa Gupta, who I think had to drop off the call. But Richa asks, how long were the EVs incubated in your uptake experiments?
So we incubated them for the uptake. For the binding, I did 2 hours. Yeah. And what I did see is that almost after 24 hours, you have almost all the cells have taken up EVs. So yeah, but 2 hours is sufficient to measure the fluorescence of the cells.
Good. Patrick James, you had a question about the mass spec.
Hi, thanks, Ken. Just look at them. Was it done by MALDI MS or ESI? The reason I'm asking is that you didn't have sialic acid content, so whether that was being suppressed.
So we measured by MALDI, and of course we did not detect any sialic acids because these parasites are not capable of generating sialic acids.
Okay. So I guess just a follow on from that, if I can, Ken. So a lot of glycans which are acylated tend to get cleared by the acyl glycoprotein receptor, but that's another uptake mechanism as well that you looked at.
Yes. So I haven't looked at that receptor, but I could definitely think, and it's a good thing that you say that, because there's actually a group in Amsterdam that looked at tumor cell-derived EVs, and also at the glycans, and that contained sialic acid. But the moment they cleaved off the sialic acids, and also, and then they saw that these EVs were also taken up more by these monocyte-derived dendritic cells, and also more via DC sign. And even when they introduced another glycan, Lewis Y, I think it was, they even saw a more increase, or at least the interaction with the DC sign. So there's definitely something happening if glycan structures on the surface of EVs lose their sialic acid.
Thank you.
Great, thanks, Pat. So we have a question here from YJohn448, but I think that this person is having a problem with their microphone. So I'm going to ask this. They write, cytokines significantly increase after adding EVs to immature DCs. I was wondering, how did you choose 24 hours for your measurement time? Would shorter time, e.g. 3 hours or 6 hours, make a difference in this cytokine release?
That's a good question. I did so normally, so it takes a while to generate enough cytokines. So normally, actually, we choose 48 hours to measure cytokines, and I just chose 24 hours, an earlier time point. But the sooner you measure the cytokines, the lower it ends up. So because we look at the ELISA, so what is released into the supernatant. But honestly, I didn't do a time check. Might be good to look into it, yeah. But I'm afraid that I probably need to add more EVs to the well, because I used quite a low concentration. I mean, I used the numbers of the NTA, but if you correspond it to protein, to the BCA measurements that I've had, I used around 1.5 micrograms per ml. So it's really low in protein content. So you actually don't add that much. It's like you have like what one parasite releases in 3 days, you give that to 10 cells. So it's not an overkill, and that is why we also had to add I1 beta and TNF alpha to get sufficient response from these cells. Because if you only add EVs alone, not much is happening. But of course, you can imagine that if you have these parasites in vivo, more directly close to the cells, and they release these EVs, you get a much higher EV per cell ratio. And of course, the parasite, the ES, these ES products are not only consisting of EVs. There are much other soluble proteins that are released that can also affect the immune system. So they probably all work together.
Good. All right. Thanks for answering that. So we now have a question from Genevieve.
Can you hear me?
Yes, we hear
you.
you.
Yeah, sorry, because I'm outside. I was just trying to figure out, are you saying that those glycans are on the filament, or are they on the surface? Are the filaments made of sugar? I'm just trying to clarify this.
So yes, that's a really good question that we're actually trying to answer now, because we hypothesized that these structures are glycans, or at least contain glycans. I can say that I visualized with cryo-EM the EVs after the PNGSF treatment, but I still saw the filaments. So the next thing is that we try to give it to optimize the protocol to also treat the EVs with a lot of proteinases to see whether it might be a protein backbone. But yeah, so we suspect that it are glycans, but the problem is that even if it would be, I mean, we're not aware of that these parasites have these long glycans strands. But also, again, if it are completely glycans, the method that we used during our glycan measurements are not able to detect these large glycans. So
actually, have you considered that these might be nucleic acid or something like that? If you think of biofilm, it's just kind of a weird idea. Since
I'm sorry, I might have missed a bit. What were you suggesting?
I was kind of wondering if it could be something like nucleic acid strands.
Oh, nucleic acid. Yeah, well,
extremely long, or maybe not extremely long, but they are long. And of course, nucleic acids are also sugar.
Yes. I haven't looked at that. So yeah, maybe. But I think we discussed it with the people from the cryo-EM facility, and normally a few strands are not visually, you're not able to see that with the cryo-EM, so it must be bigger than that, more compact, so to say. So, but yeah, who knows what we find there? It's just yeah. I hope that because I also heard from some other groups working on parasite EVs that they have seen these structures for EVs from other parasitic worms. So apparently it's not only this parasite, but also other parasite worms that have EVs with these structures. So yeah, I think we just need to collaborate and to find out what they are. Yeah.
Very interesting. Our next question is from Joy Wolfram. Hi, Joy, are you there? There you
Yes, thank you so much for the presentation. So I know that parasites can bind to hyaluronic acid. but do the EVs from the parasites themselves produce hyaluronic acid, or do they have hyaluronic acids on them because it's a major glycan?
Not that I'm aware of, because I think the hyaluronic acid it contains the I think it's also made out of these sialic acid-like structures, right? These diamond structures. So I think that kind of already makes it not likely that these parasites make such a molecule. But I haven't, so I'm not a glyco expert. That's why I have my supervisor. But I haven't come across these large glycan structures when we looked at the different, well, there are actually large glycan structures for the adult worms, but not for the schistosoma.
Thank you.
And Phil, you had a follow-up question. You wanted to know about the role of the hairs, maybe in migration.
Well, the biology, the Cercaria then migrate, I forget the exact, but they end up in the portal vein as adults. So how do these EVs with hairs participate, do you think? And now it turns out it's a general parasite thing, parasite EV, so it's very, very interesting.
Yeah, so if it's really interesting, and I think because we now also visualize the worm EVs and we don't see these hairs, so it's also apparently life stage specific, only in the larval stage. So maybe they're involved only in the initial phase, maybe indeed either targeting the immune system, maybe it's just a bystander effect, who knows, that's just beneficial. But also, I mean, the interesting thing about these larvae is they try to locate a blood vessel, but they don't go directly to the portal vein, they first go to the lungs.
Right.
And that's, I think, an evolutionary thing, because some of the soil transmitted helminths, they are also first go to the lungs and then being coughed up and then go to the gut. So it might be a remnant from that. So they go to the lungs and then eventually they go to the portal vein.
I don't think it's trivial. They had to put a lot of energy into metabolically and otherwise having these fantastic structures. So they must mean a lot.
Yeah.
In the biology of the parasite.
Yeah, that's for us to find out, right?
Yeah, it's a very significant discovery, I think.
Thanks.
So as for the visualization, Maya, you have a question about how you can see the filaments.
Hi, Marije. This was fantastic. Thank you very much. I'm working with vesicles from Trichinella spiralis. So I have, but I didn't have access to cryo EM, only to transmission electron microscopy. And I did not see such a thing, and I was wondering, is it possible to see it with the transmission electron microscopy? Have you tried?
Well, I have looked through a lot of pictures of the TEM transmission, but I haven't seen them. So I'm afraid that you really need some kind of cryo EM to be able to visualize it. Yeah.
Okay, great. Have you tried to stain them with lectins?
Not yet. No, that would be, we thought about it because of course, especially for the glycolipids, we would really like to know whether they are on the surface or not. And of course we could target that with lectins or with antibodies. But there's a lot of technical issues when you want to combine that with gold labeling and cryo-EM. You need to make sure that you have enough material that you can detect something. But I've also, I already looked into literature. Can we use some kind of staining, like for under a more advanced confocal microscope that is really sensitive? that we can do something to, because it would be awesome if we could indeed use something other than cryo-EM to measure these.
But actually, if you would use gold labeled lectins on transmission microscopy, you would see the dots, but aside from and if you do the cutting from the cellulose cuts, yeah. So I suppose you could do that.
Yeah.
Okay, thank you very much. This was great. Thank you.
Thank you.
Thank you. And it looks like we have one last question that's in the chat box, and that is from Lilia.
Yeah, thank you. That was a great talk. It's not actually a question, it's like a comment thinking to the direction that what those hairs could make a surface like for adhesion, or maybe also you've done this experiment or not, if you're taking as a host cells, not dendritic cells, but maybe some epithelial cells or something else, just, you know, look at the distribution of them and think to the direction what actually Philip asks, that what do they do in the blood circulation. So,
yes, yeah. Yes, definitely. I mean, it would be really interesting, and I've been thinking about it a lot, but that's the problem if you only have a 4-year PhD project. But of course, it would be awesome if we could investigate these EVs, especially because it's a blood circulating parasite. What did they do with endothelial cells? What did they do with all the cells in different cells in the blood? But also, if you think about the skin penetration, what do they do with the cells that they come across in the skin? So there are so many awesome things to still be discovered, and this is a really, really good suggestion, especially when it comes to these structures. What do they provide additionally? And maybe if you think about if we could dream about this generating EVs ourselves, you know, with all these fancy techniques now, can we also make these structures on the EVs eventually? If it has a benefit for maybe survival or interaction with specific cells.
yeah, thank you so much. Good luck with your journey.
So on that note, as we think about all the future things that could arise from this project, I just want to thank you, Marije, for talking with us today, for sharing your work, and for stimulating us to think about the biology of these organisms and their EVs and their interaction with the host. So thanks so much, and thanks for everybody who joined today. We really appreciate your support of the Journal Club. And I hope that many of you will be able to come to the virtual ISEB meeting and interact with Marije and many others there as well. So thanks everybody. Stay safe, stay healthy, and we look forward to seeing you next week.